Hi there. In this lecture, I'm going to go ahead and talk about the chemistry of life. Okay, so I'm going to be focusing more on what's important in terms of what constitutes, right, the main biomolecules or the molecules that make up life. Okay, so and I'm referring to carbohydrates, proteins, nucleic acids, enzymes, and lipids for that matter. Okay, so therefore, I'm going to be focusing the attention more on water, carbon, pH, and then more specifically and more importantly, polymers, monomers, carbohydrates, lipids, dietary fats, proteins, and enzymes. Okay, so starting off with a little bit of an overview of the periodic table. I'm sure everyone has had exposure to this before. Now, so several things that I want to point out here. First is the representation of the elements. Okay, so that's what the periodic table is for, right? So to be able to, for us to be able to arrange all of the elements that have been discovered, and we do so using again the periodic table and also these symbols right here. Okay, so each one of the elements contains evidently a symbol, right? For example, carbon, which is the most important element, one of the most important elements for life, is represented by C, as you probably already knew. Now the number here on top, this is the atomic number, so this is the way in which it's going to appear on the periodic table. Okay, so based off of its protons. So the atomic number is going to go ahead and represent how many protons the element contains. It's also going to represent the amount of electrons. Okay, so however many protons it contains, in this case, it's going to be 6. It's going to be the same thing for electrons. So carbon contains 6 electrons on its outer shell, okay, on its valency, if you will. That's the name of the element, and then we have the atomic weight here on the bottom. All right, so now the elements are arranged in this manner. So most of them are metals, right? So these are earth metals right here. These are transition metals. We have metalloids, and then we have the non-metals. All of these are not metals, but then these are exclusively, these are gases. Okay, naturally, those are gases. And then we have the radioactive, type of elements here on the bottom. Again, just a brief overview of what you've already seen. I'm not going to go into details because we have a lot to talk about regarding life. So we have at least 4 of those elements that you saw that are very important for life. They're crucial for life. Okay, and those 4 are going to be oxygen, carbon, hydrogen, and nitrogen. Yes, oxygen, you breathe it in, and that's important, absolutely. But it also becomes part of molecules. Okay, so it also becomes part of proteins, nucleic acids, lipids, and makes up essentially the structure, right, of those molecules. Not only does it make up the structure, but it also gives molecules some unique properties through which they're able to elicit their physiological roles. All right, so we have carbon here. Carbon is very important to make up the structure, the backbone, if you will, of molecules. And then we have hydrogen and nitrogen, which becomes part of proteins. Okay, so proteins contain in them nitrogen atoms, right? That again, nitrogen makes proteins unique in the fact that they do contain this atom right here. And I'll go over examples in a bit. There's other elements that are also required, but in smaller quantities. All right, so we're looking at calcium, phosphorus, potassium, sulfur, sodium, chlorine, magnesium, and then here on the bottom, we need these in even smaller quantities. Some of the important ones here would be fluorine, iodine, iron, and zinc for that matter. Okay, we need all of them, right? But these are important in terms of physiological function. All right. So real quick, calcium. Keep in mind, calcium becomes part of bones, becomes part of your teeth. Phosphorus is important in ATP production. Okay, so not only in ATP production, but also in nucleotide production. So nucleic acids. All right, so potassium is going to be very important in the nervous system in allowing us to carry on with producing and generating action potentials. Sulfur is part of proteins and molecules, and then sodium is going to be working right along with potassium in allowing action potentials in the nervous system to take place. Chlorine is going to be a gradient type of atom. All right, so chlorine becomes part, it's a negative atom, which becomes part of gradients. Now, these are the smaller atoms here on the bottom that we need in smaller quantities. We're looking at fluorine, which is part of teeth, right? So it becomes part of the matrix of teeth. We have iodine, which is important for thyroid function. Right? So we create two hormones specifically from the thyroid gland, which is located in the anterior neck. And the hormones are called triiodothyronine and thyroxine, or T3 and T4 for short. They require, right, 3 and 4 iodine molecules. Okay, so T3 requires 3 iodine molecules, T4 requires 4 iodine molecules to be able to be produced. Okay, so their production and their release within the body allows us to modulate our metabolism. Okay, so the reason why you have energy throughout the day is in great part because of the action of the T3 and T4, which again are made by the use of iodine. Iron is important because iron becomes part of hemoglobin. Okay, hemoglobin is a protein, rather large protein that we have in red blood cells. And at its core, it contains a heme group. Okay, heme groups contain iron in them. So that's why iron is important. And then zinc helps to modulate genes of the immune system. Okay, so you can see, right, the importance of these atoms right there. Now, a real brief review of atoms. Again, this is something you've already seen in the past. Atoms are essentially the smallest unit of matter. So all matter is made up of atoms. Okay, so up till currently, we were not able to split atoms apart. Okay, and that's essentially the definition of atoms. This is a part of matter that is irreducible, that you cannot break down any further. But it turns out that now we have particle accelerators that can actually break down atoms. Okay, and that's part of the reason why we make them. They were built actually by scientists in order to break those atoms apart and see what those atoms are made of. Okay, so again, even though that definition still applies, do know that artificially in the lab, right, we can, using particle accelerators, we can actually break down atoms. Atoms are made up of subatomic particles. Okay, so we're looking at protons, which are positively charged. We're looking at neutrons, which have no charge. These two are found inside the nucleus of the atom. Okay, not to be confused with the nucleus of the cell. Believe me, students confuse the nucleus of the atom with the nucleus of the cell. Not the same thing. Electrons are actually found outside, right? It's called the electron cloud. And these orbit the nucleus of the atom constantly. They're constantly just spinning around, orbiting the nucleus of the atom. Okay, so these are negatively charged. Let's see. So when it comes to atom weight, okay, so or atomic weight, which was one of the characteristics of the periodic table. So I showed you the number on the bottom and the symbol. That's the atomic weight. Okay, so we only take into consideration the protons and the neutrons. That's because electrons are actually about 2,000 times smaller than protons and neutrons. So we don't take those into consideration. Plus, they're always moving, right? Now, the reason why I'm mentioning that is because as atoms actually lose or gain electrons, they become something called ions. Okay, so in losing electrons, naturally you become more positive, right? So if an atom loses an electron, it's going to become one electron. It's going to become in a cation, which has a charge of +1. If it gains an electron, then it becomes an anion, which has a -1 charge. Okay, so again, losing any electrons makes atoms ions. Now, in actually making molecules and things that are larger, then you have to deal with the transfer of electrons and the sharing of electrons between atoms. Okay, because that's the only way in which we can actually make larger molecules and larger organisms, for that matter. So we have to deal with bonding. There's a few ways in which atoms can bind these essentially to each other. We have ionic bonding, which is defined as a basic transfer of electrons from one to another atom and vice versa. So these two atoms right here are going to be sharing electrons constantly, right, at all times. And as long as those electrons are flowing from one to the other, then we can call this an ionic bond. Okay, so we're looking at sodium here, chloride here. Both put together, we make sodium chloride, which is your table salt. Now, the other type of bond is covalent bonding. So this deals with the sharing of electrons, right? Sharing of electrons, whereas the other one was the transfer of electrons. Now, we have an equal sharing of electrons, but we can also have a non-equal sharing of electrons. All right, so if atoms share electrons equally, then we call that a nonpolar covalent bond. Okay, so these 5 atoms right here, we have a carbon atom in the middle. Excuse me. We have hydrogen, 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 and hydrogen right there. So 1 carbon, 5 hydrogens sharing electrons equally. Okay, so that again, that's called the nonpolar covalent bond. Then we get into the polar covalent bonds, which is a non-equal sharing of electrons, such as these 3 atoms in a water molecule. We have an oxygen in the middle, we have a hydrogen, and another hydrogen right outside. So electrons are being shared, but they're being shared unequally, so unevenly, if you will. Right, so again, polar covalent bonding. Now, water molecules can form hydrogen bonds, right? And that's essentially the fact that gives water its crucial properties, like cohesion and adhesion, okay? So again, hydrogen bonding between water molecules. You're looking at a water molecule here in the middle, and then another water, 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 and water. So there's 5 water molecules right here, all trying to come together through hydrogen bonding. Okay, so the reason why you see those water molecules in a cup of water, right, staying together is because of these hydrogen bonds. As soon as you break those hydrogen bonds, for example, when you add heat, right, you evaporate that water. So those water molecules start to diffuse, right? They start to become apart from each other, or depart from each other, rather. But again, that's because you added a lot of heat. You need to add a lot of heat in order to break these hydrogen bonds. Okay, but if you don't add heat, then they'll just naturally come together and form the bonds that essentially make water the liquid substance that you know that you see and you drink essentially every day. Okay, and you shower with every day. So here's more or less how it happens. You have a negative oxygen, you have a positive hydrogen, right? How do you know they're negative and positive? We'll just look at the periodic table, right? So oxygen is going to be negative, hydrogen is going to be positive, so their opposites attract and they come together. Same thing here. Now look at the hydrogen from this central water molecule. It's positive, right? So it's not going to attract the hydrogen because the hydrogen is positive. So it attracts the negative oxygen from another water molecule so that it can actually come together. Okay, so that keeps on happening all throughout. In this case, this water molecule is already saturated, but if you picture more water molecules on this side, then these are going to go ahead and bind to this water molecule. If you picture more, then these bind to this water molecule, and so on and so on. Okay, so there's more properties for water. For example, the bond angle right here. Again, polarity, right? So meaning that water is a polar molecule, meaning that its atoms share electrons unevenly, right? Essentially forcing that oxygen to keep most of the electrons close to its core at all times. Okay, not that hydrogens don't get a share of the electrons, they do, right? But they're gonna go and do so in less quantities, if you will. Okay, so now another property of water that's very important, and it is due to the fact that it is a polar substance, is the fact that water is actually a solvent. Okay, so water can dissolve a lot of molecules, a lot of solutes. Okay, so that's essentially what I mean by water being a solvent. Okay, so it's a very successful solvent, so successful that it's actually known as the universal solvent. Okay, so as long as the solutes are polar, then they can go ahead and dissolve in water. Now, if the solutes are non-polar, then they cannot dissolve in water. Okay, because again, of the differences in properties in terms of sharing of electrons. Okay, they're different. For example, if you add salt into this cup of water right here, will it dissolve over a period of a few minutes? Absolutely. Okay, so over a period of a few minutes, these salt molecules here are going to go ahead and completely dissolve and become embedded within the network of water molecules right there. All right, so now if you add in more sodium, if you saturate the solution, then any extra sodium chloride is going to actually precipitate out of the solution. But by that time, again, you would have already saturated all of the water molecules right there. Now, if you add, instead of salt, if you add oil to the water, will it dissolve? Evidently not. And you probably tried that before. You know that, for example, when you're washing dishes, right? You know that you can't you can't expect to get all that grease out of the pans, right? Only with water. You can't. You're not gonna be able. You're gonna be able to achieve that to a certain extent as long as you're scrubbing that pan, right? But in case you didn't know, there's no need to scrub pans anymore because our detergents actually take care of the dissolution of those greases there. So all you got to do is just fill the pan with water, add detergent, right? And then just lightly scrub that grease, right? So what's happening there is that the detergent is actually binding, right? It's attaching to those grease molecules. Detergents are amphipathic molecules, meaning that they have a polar and a non-polar side. Okay, so that's why detergents are crucial in dissolving fats and greases, if you will. All right, so they'll attach to the fat and they'll dissolve the fat readily. All right, so no, water is another property of water is that it can absorb and store large amounts of heat while it itself not changing so much in terms of the degrees that it's gonna go ahead and amount. All right, so it's a great modulator of temperature. And that's why water, right, in our blood is actually in charge. One of its roles is to modulate the temperature inside of our bodies, right? What happens when we get hot enough? Well, we start sweating, right? So that sweat, those sweat glands actually start um, releasing water onto the surface of the skin, which has an evaporative cooling effect. All right, so same thing happens throughout the earth, right? So those oceans, in case you didn't know, they have a big, huge role in modulating the temperature throughout the earth. Okay, why is that? Well, because oh look, I'm showing an example of evaporative cooling. Right? So you can see this player, or you can imagine, right? Visualize this player being highly active in the game. Right now, he is in the bench, obviously, and you can tell that the environment is pretty cold, right? Because he's evaporating that or dissipating that heat readily. So again, the water that is released onto the surface of our skin manages to, and it's actually enough um, so that we can dissipate most of the heat that is created inside of our cells, inside of our bodies, due to our high metabolism. All right, so other properties of water are cohesion and adhesion. Cohesion meaning the tendency of water molecules to stick together, and then cohesion, those water molecules sticking to the surfaces of their container, essentially. Okay, so very important properties, especially when it comes to circulating water around the body, right? So water doesn't exist as pure water inside the body. Obviously, it's dissolved in solutes, right? Like for example, blood that contains red blood cells, white blood cells. It contains enzymes, proteins, lipids, and so on. Right? These cohesive properties of water allow it to stick to the walls of our blood vessels, and therefore they're able to move through the blood vessels pretty readily. All right. So another property of water and characteristic of water is the fact that it can regulate pH. Okay, it can especially in the body, right? Our blood has to be kept at a pH of about 7.35 and 7.45, give or take. All right, so that's more or less where the blood has to be, pH has to be kept. So water has a big role in maintaining that pH, but it does so through the use of blood and through the use of something called carbonic acid. All right, so carbonic acid it has the ability to either release hydrogen ions into the blood or actually capture and trap hydrogen ions from the blood so that they can become carbonic acid. All right, that's called a buffer, right there. That's a buffer system. Okay, so buffer. Cells actually regulate their own pH through the use of buffers, and one of the main buffers inside of the body is, again, carbonic acid. All right, so if you recall the pH scale, it goes from 0 to 14, 7 being neutral, and then anywhere below 7 is acidic, anywhere above 7 is basic or alkaline, right? So again, keeping that pH constant at 7.4 in the body takes the use of buffers. In our case, carbonic acid. All right, so carbon is, before I get into the biomolecules, carbon is a very important atom in the body, which takes care of essentially the backbone. It becomes the backbone, right, of molecules such as proteins, nucleic acids, lipids, enzymes, and so on. All right, so carbon's important because it can actually form up to four bonds. They can be single bonds, right? Such as the ones that you're looking at here. These are single carbon bonds, right? And if you have one carbon atom in the middle, then it can actually fit four atoms right around it. Okay, so this carbon can fit four atoms around it. 1, 2, 3, 4. But the thing is, those bonds can actually become a little bit sophisticated, right? In terms of the arrangement of the bonds. So you have a linear carbon chain right here. You have a carbon chain that is branched, right? And then you have carbon chains that are actually forming rings right here. Okay, so very important in making your proteins, your amino acids, your carbohydrates, right? For example, this branched carbon chain here is important in the making of carbohydrates, for example. Okay, so as long as you add some oxygens throughout, then you'll have in the right amounts and arrangements for that matter, then you'll have a carbohydrate here. These ring arrangements are important in making steroids. All right, so steroids like testosterone and estrogen, right, which are the female and male reproductive hormones. All right, they fall under a category called steroids, and those are mostly made up of carbon rings. Right, steroids contain usually 4 carbon rings to them. So again, just highlighting the importance and differences between the arrangement of bonds and their branching patterns. So if you utilize other atoms like oxygen, like nitrogen, right, like phosphorus there, and you put those atoms together with carbon, then you make organic molecules. Organic molecules tend to have carbon atoms at their core and as part of their backbones. All right, so if you add one oxygen atom, right, single bonded to a carbon atom, then you're going to make something called a hydroxyl group, which becomes important in the making of lipids and carbohydrates. If you add a nitrogen to single bonded to a carbon, then you make an amino group, which is important in creating amino acids that are going to eventually make up proteins, right? If you add phosphorus to 4 oxygens, or rather 4 oxygens to 1 phosphorus, then you make highly energetic bonds right here that can release a lot of energy, a lot of electrons. Okay, so we're looking at phosphate right here. All right, so now in making these large molecules from smaller molecules, right? So we get into the topic of monomers and polymers. All right, so monomers are essentially smaller parts of polymers. Polymers are made up of monomers. Okay, so hopefully that makes a little bit of sense. Let me go into a little bit more detail here. If you were if you were to put together all of these amino acids here, how do you know they're amino acids? Well, because they have nitrogen. So put them together in linearly, right? So one after the other, using peptide bonds. So well, all of a sudden you made a polymer made up of 4 amino acids held together by peptide bonds. And again, each individual molecule here is known as a monomer, whereas a whole thing put together is known as a polymer. Okay, so this is an example, right? Proteins are polymers because they're made up of hundreds of amino acid subunits, right? But nucleic acids are also polymers because they're made up of hundreds of nucleotide molecules put together. Okay, so carbohydrates can also be polymers and so on. All right, so now in breaking down a polymer, which happens essentially at all times, right now as we speak, right? You are breaking down polymers, right, in your cells. Due to the fact that they're probably expired, they're older, right? You need to break them down so that you can rebuild those amino acids. Then what you're going to do, what your cells are going to do is they're going to go ahead and hydrolyze these big molecules by adding water to them. So they add water to them. That's called a hydrolysis reaction, which takes care of the breaking down of those big molecules into their constituent molecules, into their monomers. So in that sense, you can break a polymer down into its monomers, right, through hydrolysis reactions. Now, on the contrary, you can build large polymers by putting together monomers. Okay, so how do you do that? By dehydration reactions. So bringing these two, for example, these two together, right, getting rid of this OH and this H molecule right there, that makes water, right? So you dehydrate those two molecules, and then in that sense, they can go ahead and become, attached to each other. Again, dehydration reactions makes up large polymers from smaller monomers. And that's taking place, that's occurring at all times in every single one of our cells. Alrighty, so let's get into the biomolecules. Okay, we're talking about carbohydrates, lipids, nucleic acids, and enzymes. Okay, so let's get into it here real quick, starting off with the carbohydrates. These are sources of carbohydrates. Okay, we're talking about bread, potatoes, pasta, rice, and so on. Right? All of those food sources contain carbohydrates. All right, so now carbohydrates, we have simple carbohydrates, we have complex carbohydrates, but we're going to go ahead and give them these names, okay, monosaccharides for simple carbohydrates. So one glucose molecule, for example, is a monosaccharide. Okay, one fructose molecule is a monosaccharide. Okay, so glucose, fructose. Notice the similarities. So these are highly similar molecules right here. Right, the only thing that differs from them is their arrangements. All right, so now even though they're essentially the same thing, the same molecule, they're isomers because their atoms have different arrangements. Notice how the formulas here are essentially the same, right? So you wouldn't be able to tell them apart just by writing the formula down. You would need to look at the structure of them. All right, so now again, simple sugars are monosaccharides. Okay, so monosaccharides, if they come together, if you have enough monosaccharides as monomers coming together, then you make polymers of carbohydrates, right? So their attachment, their bonding right there is going to go ahead and be called a glycosidic linkage. So this right here is called a glycosidic linkage, right? And it was made by the release, right, of water. So what type of reaction brought these two monosaccharides together, right? Was it hydrolysis or was it dehydration, right? So keep in mind, releasing of water, right? So you release that water molecule right there. That's a dehydration reaction, which puts together smaller molecules to make up larger molecules right there. So all of a sudden, by putting glucose and fructose together, you make sucrose. All right, so for the most part in biology, right, if the word ends in O's, then it's probably a carbohydrate. Right? That's another little characteristic of carbohydrates. So animals use carbohydrates, plants use carbohydrates, and they store carbohydrates in the form of starch, right, plants. So you're looking at potatoes right there with storing, right, carbohydrates as starch. There's also cellulose. Cellulose becomes part of the cell walls of plants. All right, so that's another type of carbohydrate, complex carbohydrate there. Animals utilize carbohydrates in different ways. Okay, so we store glucose, for example, in a large molecule called glycogen. Glycogen is found within the liver, and it's also found around the muscles, within and around muscles. All right, so why is glycogen important for us? Well, because it allows us to store potential energy so that we can rely on that later on, okay, throughout the day, throughout our life. All right, so the more carbohydrates you ingest, then the more glycogen you're gonna go ahead and form, unless obviously you utilize it to a high degree, right? Then you can go ahead and essentially run out of your glycogen stores. But that's because you're using that those glycogen stores, such as this athlete right here, training, right? So another way in which animals use carbohydrates is to, for example, to build an exoskeleton. Right? The exoskeleton of this animal here, this insect, is made up of a carbohydrate called chitin. Okay, so again, it's a rather tough structure that not only provides structure to the organism, but it also gives it protection, essentially. Okay, so all right, so now let's talk about fats here real quick. Fats are also known as lipids. Okay, so we actually call them lipids in the medical field and in the biological sciences. Okay, so we refer to fats as lipids. Functions of lipids are to store energy, to cushion, to insulate. They make up membranes and they regulate hormones. And by the way, the function of carbohydrates is to provide a quick source of energy. Okay, so let's go over those here real quick. Lipids are diverse groups of organic compounds because they do contain carbon at their core. Lipids are mostly phobic, whereas carbohydrates are actually hydrophilic. So most carbohydrates are going to dissolve readily in water, whereas lipids are not going to dissolve at all in water, unless, of course, you add a detergent. Okay, so now part of the reason why lipids are important in the body is because they make up the membranes of all of our cells, the trillions of cells that you are made up of. All of them contain a cell membrane made up of phospholipids. Okay, so here's more or less what a phospholipid looks like. It contains a phosphate head and lipid tails. All right, the phosphate head is in and of itself, it's a hydrophilic substance, whereas the lipid tails are evidently hydrophobic. Okay, so why is that important? Well, because as you're putting together these membranes, okay, you're being essentially two layers of phospholipids, right, which are inverted. And those two layers are actually called a phospholipid bilayer. Okay, so now the reason why they're inverted like that is because you want these lipid tails to face the inside of the membrane, right? They cannot interact with water because they're hydrophobic. Therefore, you want that phosphate head to be interacting with water while keeping that lipid tail inside of the membrane right there. All right, so just key details about the structure of membranes and some of the properties of membranes as well. Like for example, the fact that membranes have embedded proteins. So these proteins are crucial in the transport of molecules from the outside of the cell to the inside, and from the inside of the cell to the outside. Because water molecules move in and out, sodium ions, chloride ions move in and out through these transmembrane proteins right there. Okay, so membranes are also composed of cholesterol molecules. Okay, so these right here would be cholesterol molecules, which are essentially steroids. All right, so cholesterol are complex fats that make up not only membranes but also hormones like testosterone and estrogen. Okay, so that's why cholesterol is important. Then the thing is, cholesterol is transported in the blood, right? So if you have an excess of cholesterol, you can actually build up that cholesterol in the blood, which becomes embedded onto the walls of the blood vessels, right? So if that happens, then you run into an issue of generating obstructions to blood vessels, which are going to go ahead and slow down blood flow, right? So if blood flow slows down, then the heart has to compensate for that by accelerating, so it beats faster, so that it can make up for the loss of blood flow at a certain point within the blood vessels, right, within the circulation. So that accelerated heart rate, if it's chronic, if it's over time, then evidently that's going to lead to cardiovascular disease, right? So all of a sudden you would see that person, right, with a chronic high heart rate. You would see that person with an enlarged heart, right? So the heart is a muscle, right? And you can work out that muscle, right? So if you make it work harder at all times, right, for an elongated period of time, then it's going to grow. It's going to become enlarged, right, which is pretty dangerous for for a person. So this molecule right here, it's got 4 rings, right? It's 4 carbon rings, 1, 2, 3, 4. This becomes a steroid. Okay, it becomes a steroid molecule, which again is important for making generating testosterone and generating estrogen in males and females. So cholesterol is very important. Lipids are very important for the functioning, right? For the optimal functioning of the organism, right? In this case, again, cholesterol has a hormonal regulation type of role here, but lipids are also part of membranes, right? And then lipids also become important in, for example, animals that hibernate, right? Animals that hibernate in places that are very cold, like polar bears. Well, in the time they're awake and active, what they do, well, they look for food and they're eating all the time. Okay, they get big, they get fat, so that during the months that they're hibernating, there's no need for them to go out and look for food, right? So they've already collected enough fat in their bodies, and all they have to do is just go rest and sleep it off for a few months, and they rely on their stores of fats, right, to stay alive. Okay, so now let me go back to the discussion on the circulatory system and fats within the circulatory system. So we have 2 types of proteins that actually transport cholesterol. Okay, so we have low density lipoprotein, LDL, or in high density lipoprotein, HDL. So LDL would be the bad cholesterol, HDL would be the good cholesterol. Okay, this is the good cholesterol because HDL actually captures cholesterol from your blood and takes it over to the cells where it's going to go ahead and be used for, for important cellular physiological purposes. Okay, LDL is going to do the opposite. It's going to take that cholesterol back into the blood, right? So which is okay because you want some cholesterol in the blood so that it can be transported to places, to tissues in the body where cholesterol is needed, right? But the thing is, have more than enough LDL, then you're gonna have more than enough cholesterol in the blood, if that makes a little bit of sense there. All right, so having a poor diet, for example, increases your low density lipoprotein. If you have an increased low density lipoprotein, then you're most likely going to have cholesterol in your blood more than is required, right? With, again, coming back to cholesterol producing obstructions in the blood vessels, right? It's going to go ahead and become embedded onto the walls of the blood vessels, and then ultimately making a lump, right? Those are called plaques. Okay, plaques are made up of mostly fats and cholesterol. So they collect throughout the blood vessels, and again, they make the heart pump faster because all of a sudden, blood is actually facing these obstructions, right, which are slowing down blood flow. So these are some sources of good cholesterol right here. So we're looking at fish, meat, we're looking at red meat, eggs, beans. So these 2 right here, fish and beans, would be the more reliable cholesterol and protein for that matter source. Okay, so the reason why I don't recommend red meats is because red meats come from highly metabolic animals. Okay, so then those animals are actually carrying on with cell respiration at a very high rate, which releases carbon dioxide and other toxins into their tissues, right? So if these animals are killed for food purposes, right? They are killed with a very high amount of carbon dioxide and toxins in the meat in their muscles, right? The muscles become meat that we actually ingest. So we're actually ingesting a high amount of CO2, a high amount of toxins came from the metabolism of the animal. All right, so yes, it's okay to eat meat, but maybe once a week, maybe once every red meat, once a week, maybe once every 2 weeks would be ideal. But eating red meat every day or every 2 to 3 days, well, that's gonna take a toll on your body eventually. Okay, so these two right here, these have fish has a very low metabolic waste output, right? So that's why fish is essentially healthier in terms of consuming, right? And then beans, obviously, these come from plants, right? So these have very, very low amounts of toxins, very low amounts of waste products here. So, it would be the purest, right? The purest type of protein source that you can actually find, okay? Okay, so now lipids are actually made up by these molecules called triglycerides. Okay, so triglycerides are at the molecular level, they're carbon hydrogen chains. All right, so we're looking at a triglyceride right here. Okay, so this part of the molecule is called glycerol. All right, so glycerol is made up of a 3-carbon chain with oxygens, right, attached to those 3 carbons. And then the tri part of the word comes from the 3 lipid chains, okay, that you're looking at here. So again, this collectively is known as one triglycerol molecule right here, which makes up most of your fats. Okay, so now, again, I've already talked about steroids. Steroids are made up of, at their core, they're made up of cholesterol molecules, which have 4 carbon rings to them. Steroids are important because of their function in making sex hormones like testosterone and estrogen, right? So look at the similarities between these hormones right here. They're very similar, with the exceptions of a few differences in terms of, for example, this oxygen here, right? This oxygen is double bonded to this ring, right? Whereas this oxygen is actually single bonded to the ring, and then it contains a hydrogen, right? So this is a hydroxyl group, and then this is a conyl group right here. So basic differences between testosterone and estrogen, they're almost the same thing. One of them is found mostly in males, so testosterone, and then estrogen is found mostly, evidently, in females. What do? Well, they're responsible for primary and secondary sexual characteristics of animals. So getting to just kind of extending the topic of steroids, right? There's these drugs that are called anabolic steroids. Okay, so ingesting those can actually increase performance of athletes. Okay, it can increase your performance, whether you're an athlete or not, right? Then the thing is, they have consequences. They have secondary effects that are obviously not. I'm sorry about the noise. I'm in my garage right now, so you're gonna, hear some weird noises in the background. Sorry about that. So again, anabolic steroids help in the synthesis, right, of testosterone, But here's the thing: anabolic steroids are actually analogs of testosterone, and they look like testosterone. So therefore, injecting them, right? So putting them into the body in the form of, for example, THG, which is used to be a very common anabolic steroid. So over time, right, if athletes are doing that very often for a prolonged period of time, then they're gonna go ahead and make the natural ability of our body to produce testosterone. They're gonna go ahead and lower that, right? The body all of a sudden starts to shut down in terms of creating natural testosterone because it doesn't feel the need to, because you're putting in testosterone, right? So we have this type of hormonal regulatory mechanism called the negative feedback loop. A negative feedback loop starts in the hypothalamus. It includes the pituitary gland and also the organs, right? So the feedback loop of testosterone and estrogen, for that matter, takes a hormone, a hypothalamic hormone called gonadotropin releasing hormone, which stimulates the pituitary gland to produce FSH and LH. So those two, they travel to the ovaries and testes via the blood to stimulate the production of testosterone or estrogen, depending on, obviously, whether you're female or male. And then the production of those two hormones actually communicates back to the brain, to the hypothalamus, and tells the hypothalamus to stop the production of FSH, or actually GnRH, because there's enough already testosterone or estrogen in the blood, right? So again, that's a negative feedback loop. So putting in THG into the body is actually inhibiting the production of GnRH essentially at all times, right? And that's where the problems come in, right? Over time, these athletes stop their natural production of GnRH, FSH, LH, and testosterone or estrogen, and they can actually damage their bodies, right? They can suffer from the underproduction of those hormones. All of a sudden, if they stop using, right, their anabolic steroids, so they can become essentially in a way addicted, right, to the steroids, because their bodies are actually asking for them over a prolonged period of time, right? So again, it takes time. It takes a constant use of these steroids. They used to be legal at some point, right, when they were discovered, but then obviously over time they became illegal because these athletes were becoming way too good, right, at running or football athletes or baseball athletes, such as the one that we saw right in that picture. So we can classify fats into different groups. So we have saturated fats, we have unsaturated fats, and then within unsaturated fats, we have trans fats and healthy fats. Okay, trans fats, if I'm not mistaken, they should be banned. Okay, we actually reduce the amount of trans fats that were released to grocery stores because they are dangerous. Okay, they collect in the blood vessels and they cause heart attacks. So healthy fats, evidently, right? Normal fats are considered healthy because they become part of the physiological processes that we actually need them for, like insulation, right? Like making membranes, like cholesterol, testosterone, estrogen, and so on. All right, so now saturated fats are saturated because their carbon chains are full with hydrogens. Okay, so they are full with hydrogens. You can assume there's an oxygen here on this side, or maybe another hydrogen, or maybe a glycerol molecule. Right? So they're full with hydrogens. That's why they're saturated. Unsaturated fats are not full with hydrogens. They actually have double bonds. Okay, so they have double bonds, which takes out the ability of that carbon chain to be saturated, to be full with fat. All right, so some characteristics of saturated fats are that they contain the maximum amount of hydrogens that they can contain. They're solid at room temperature, right? Like your margarines, for example. And animal products have a higher amount of saturated fats. They have all single bonds and they're linear in shape, okay? Which makes them a little bit less healthy than unsaturated fats. Okay, so these triglycerides, for example, are known as unsaturated fats because they have double bonds to them, which means that they're not going to contain the maximum amount of hydrogens that they can contain because of the double bonds right there. Okay, so and for the most part, they're gonna be liquid in room temperature. Okay, so liquid at room temperature. You find unsaturated fats mostly from plant products. Okay, so like oils, for example, I believe canola oil, vegetable oil, obviously. And they're mostly going to have a bent shape due to the double bonds that they contain. So let's see, fats. Some of the healthier fats that we need to consume are called omega fatty acids, okay, like omega-3 fatty acids. Okay, they have a more healthy, right, origin, like chicken, like lean meats, like chicken, um, beans, obviously eggs, right, fish, and so on. So let's move on to proteins. Proteins are very important in the fact of they have a high physiological importance. That's what I mean. Okay. Now proteins come in many different forms. They have many different shapes, and they actually have certain levels of folding. Okay, so we make proteins by folding these long linear polypeptide chains. Okay, so those polypeptide chains come from obviously cells. They're made in cells, and they are chains of amino acids. Okay, so there's another group of molecules that are also known as proteins, right? They're the enzymes. Enzymes are proteins. They're the workers, if you will, of the cell. Okay, so again, proteins are highly diverse. They have their unique structures, their shapes, and these are the sources of proteins, right? So meats, um, fish, beans, eggs, and so on. Okay, so now proteins are large polymers made up of hundreds of monomers. Okay, so the large polymer is called a protein, where are the smaller monomers called an amino acid for proteins. This is more or less what an amino acid looks like. Amino acids are carbon chains with nitrogens in them and a couple of oxygens as well. So this group right here is called an amino group, and this group is called a carboxyl group. So if a molecule contains an amino group and a carboxyl group on the same molecule, then that's an amino acid. And you can actually form chains of them to be able to form polypeptide chains, also known as proteins. How do you put them together? How do your cells put them together? Well, by dehydration reactions, which take out water molecules in order to make larger molecules. All right, so we're looking here at a folded protein, but then with an extension here, it's polypeptide. So you can say this is the primary structure of that protein, right? It's called a polypeptide chain. It's a linear, obviously, chain made up of many hundreds, right, of amino acids. Okay, so each one of these spheres is an amino acid, and then these little strings right there that put the amino acids together, those are called peptide bonds. All right, so now this is the way in which we originally make proteins. We make linear chains of these amino acids. But then ultimately, they have to be folded into their secondary chain, secondary structure tertiary structure, and then ultimately quaternary structure. So that ultimately you can make up a functional, physiologically functional protein. So as mentioned, proteins have a wide array of functions, of roles in the body. Okay, so they're essentially the workers of the cell. They do everything for the cell. Some examples are, for example, structure. For example, keratin, right? Keratin makes up the main protein that our hair, for example, is made up of. It's also found on the skin. Proteins are enzymes, right? Enzymes are the little workers of the cell which speed up chemical reactions inside of cells. Without enzymes, there wouldn't be life. Okay, so without enzymes, you wouldn't have ATP. You wouldn't be able to make ATP in time. Okay, so enzymes again speed up chemical reactions, especially metabolic reactions inside of your cells, which allow you to make ATP a lot of ATP in a rather short amount of time. Okay, so there's also movement. The muscle contraction molecules are actin and myosin. Those are proteins. Proteins also become part of the immune system, right? They become antibodies which fight off infections, right? Bacterial and viral infections. Proteins have also other key roles. For example, in making up these proteins called hemoglobin, right? Which can transport oxygen throughout the body. So the shape of the protein has a very important. It has a key role in determining the function, right, of the protein itself. Proteins have to have their unique shape. They have to be folded into the right structure in order to be considered a normal protein. Sometimes there's mutations, right, which lead to a change in the shape of the protein. Okay, so those mutated proteins, obviously, they're not going to have their normal physiological functioning, right? And here's one example. We have round spherical red blood cells with their biconcave shapes, but then in sickle cell anemia, these red blood cells became sickle shaped, right? Because of that hemoglobin being mutated. Okay, so again, this one example of the importance of having the right amount of amino acids, having the right structure, right, of the protein there. As mentioned, proteins are also enzymes, right? Enzymes speed up chemical reactions in the body, and these are more importantly in metabolism. Okay, metabolism is that physiological aspect of the organism that allows you to become active and further make ATP, make a lot of energy, right? So that you can sustain your everyday activities, walking, running, your sport, thinking, right, going to school, and so on. So again, that takes a high metabolism. Athletes, for the most part, tend to have a really high metabolism. And again, that's due to the fact that their enzymes, metabolic enzymes, are optimal, right? So they have to maintain a rigorous type of diet, which would be the normal diet that the human is supposed to have, right? If you think back, thousands of years ago in the early humans, right? What did they eat? They didn't have chips, right? They didn't have processed foods like we have nowadays. So they used to eat obviously mostly vegetables, right? Mostly fruits and vegetables, mostly plant-derived foods, and then with the occasional meat, right? And I'm saying occasional because hunting took a lot of energy, took a lot of time. So if they had a piece of meat every 2 weeks, they were lucky, right? So maybe every week, right? Because of that they had a successful hunt, if you will, then they would consume right that meat. But for the most part, mostly their diet consisted of plant-based products, which is true for athletes nowadays. Okay, so again, that keeps their enzymes optimal. And here's what I'm showing here is a functioning of an enzyme. Okay, so you have an enzyme right here. Enzymes can contain two sites. They contain an active site and they contain an allosteric site right here. The active site is for the activation of the enzyme. So making the enzyme work takes the substrate right here, binding to the active site, and that activates the enzyme essentially. Okay. Now the allosteric site, that's more for regulation of the enzyme. So yes, you want the enzyme active, but you have to regulate it. You have to downregulate it also, okay? Because having an active enzyme all the time is gonna eventually lead to exhaustion problems. Okay, so now here's that regulation that I was talking about, right? So the regulation is gonna go ahead and take several shapes. Okay, it can take the competing for the active site. So if you have a substrate here, which looks similar to the regular substrate, but it's not the ideal substrate, then it's going to be a competitor, right, to that substrate because it's going to go ahead and fight for the active site of the enzyme. So that's one regulatory mechanism. Another regulatory mechanism, as mentioned, was the allosteric site. Okay, so you have the non-competitive inhibitor because it's not competing for the active site. It doesn't care about the active site. It cares about the allosteric site. So it binds to the allosteric site. It's going to change the shape of the active site, therefore not allowing the substrate to bind to the active site anymore because it doesn't have the same shape, right? So the substrate can't bind to it anymore. And in that manner, right, it deactivates that enzyme to a certain extent. Now, if the cell needs the enzyme again, then all it's got to do is take that non-competitive inhibitor out of the way, right, out of the allosteric site, so that the substrate can bind to it again due to the reverting, if you will, of the shape of the active site. All right, so I'm just kind of elaborating here on what I've already said. Enzymes require the right shape, right? Substrates require the unique shape so that they can actually bind to the enzyme and activate that enzyme. Okay, so with that said, that was the last concept of this lecture. Again, this lecture was on the chemistry of life. Biomolecules, right? So I talked about structures and functions of the three types, I'm sorry, of biomolecules. I didn't mention nucleic acids because we have a whole unit on nucleic acids, but do know that nucleic acids are yet another type of biomolecule. All right, so if you have any questions, please be sure to email me those questions or maybe request a virtual office meeting.